Hello guys, so in this video we're going to talk about nucleic acids. We're going to go from structure to function. We're going to talk about DNA and RNA and how their function ultimately is to store information and then use that information to produce proteins. Okay, so let's begin. The nucleic acid is a polymer. Okay, so just like the protein is a polymer and it's made up of the monomer, which is amino acids, we've got polysaccharides, which are the polymer, and they're made up of the monomer, that is monosaccharides. The nucleic acid is a polymer and its monomer is the nucleotide. Okay, so let's have a look at the structure of that. Alright guys, so here I've just drawn our three components of a nucleotide. First of all, here is our phosphate group. Okay, it's our phosphate group. Or it's a phosphate molecule at the moment. This is our pentose sugar. Now, in this case, it's deoxyribose, but and that's in DNA. But in RNA, this pentose sugar would be different. It would be ribose. Okay. Right. So we will. Okay. Right. So this is our pentose sugar, and here we have one of our 5 different possible bases or nitrogenous bases. In this case, I have here thymine. Okay, so thymine is the example that I've got here, but in a general nucleotide, we would call it a nitrogenous base. Okay, so 3 components of a nucleotide: pentose sugar, a phosphate molecule, and a nitrogenous base. In this case, I've got thymine. It could be one of 4 others. Okay, yeah, we'll discuss the specifics of bases and their combinations later. Okay, so how do these come together to form the nucleotide? Luckily, it's nothing new here. It's our old friend, the condensation reaction. Okay, so just looking at this diagram, you'll be able to see how it's going to be connected through condensation reaction. Very simply, 2 hydrogens and an oxygen there. That's going to form a water molecule. And similarly over here, 2 hydrogens and an oxygen there, and that is also going to form a water molecule. When that happens, the remaining atoms, which all have lost 1 bond, they will satisfy their need for a covalent bond by bonding to each other. So in this case, the carbon to the nitrogen, and in this case, the oxygen to the phosphorus atom. Okay? Right, so let's just have a look at what that would look like. So we've lost the OH there, and the H from that O, that's not there anymore. And we've lost all of that because that has now become the water molecule there. So the new bond which is formed is there. Bond lengths not to scale. And there, from the carbon to the nitrogen. And thus we have a nucleotide. Okay, so let's just quickly talk about the new bonds that are formed. Here, the bond between the phosphate and the sugar is called an ester bond. Okay? And the bond between a glucose and another group is called a glycosidic. Okay, so here we have an ester bond, and here we have a glycosidic bond. Okay, and thus we have the three components of our nucleotide. In all the following discussion, I'm going to simplify this right down to a much simpler organization, just to keep us sane. I'll denote the phosphate group with a circle. I'll have a kind of pentagonal shape for the pentose, and I'll just draw the base with a kind of square shape. Okay, so there we have it. I'll rejoin you now when we discuss how these nucleotides can be joined together to form the polymer. Okay, remember, this is our repeating unit, the nucleotide, and this is going to be then joined again and again to form the polymer. Right? And the polymer can be either DNA or RNA, but they're both made of nucleotides. Okay, see you in a bit. Right, welcome back. Right, so we discussed the detailed structure of the nucleotide just now. I'm going to simplify that now to something like this, because as our polymer grows, it's going to become a bit more complex, and it's not going to be convenient to draw all the atoms out. Okay, so here we have the same thing that you've just seen, just simplified. Phosphate group attached by the ester bond to the pentose sugar. It could be a ribose or deoxyribose. Pentose sugar attached to the nitrogenous base by a glycosidic bond. Okay? Now, what happens next is in the next level of structure, so moving on from the monomer, what can happen is different nucleotides can line up and be joined together. But this is not the same case as a carbohydrate. In the carbohydrate, you remember that it's always the same repeating unit joined end to end. But in the case of nucleic acids, you don't have the same nucleotide over and over again. You have different versions of that nucleotide, and what makes the different versions of the nucleotide is not the phosphate or the sugar. They're always the same, but what you can have is different bases, okay, nitrogenous bases. Now there are 4 nitrogenous bases, so let's just not get ourselves confused. The sugar can be of 2 types. If it's DNA, that sugar is going to be deoxyribose, always. If it's an RNA nucleotide, that sugar is always going to be ribose. Okay, now let's look at the bases. The bases are like this. There are 4 bases in each of these. Now, in total, there's 5 bases. In DNA, there is adenine, thymine, guanine, and cytosine. Okay? However, in RNA, you don't have thymine, you have uracil. So what I'll do is I'll just put a bracket in RNA, uracil, not thymine. Okay, so in total there are 1, 2, 3, 4, 5 bases, but all DNA nucleotides will have the deoxyribose sugar and these 4 bases, and in RNA the nucleotides will be phosphate and ribose sugar plus adenine or guanine or cytosine or uracil, not thymine. Okay, so this is the situation, and we'll move on now. Okay. Right, so how does the polymer form? Well, you recall that these carbons had OH groups on them, and one OH group was used to form the ester bond to this phosphate. Now, if we have another nucleotide lining up down here with its own nitrogenous base, now let's get a bit more specific here. Let's say in this example, we're looking at a chain of DNA nucleotides. The first one might have contained the nitrogenous base adenine. The next one might contain the nitrogenous base guanine, okay, for example. Now what happens is that this phosphate group can form an ester bond with one of the OH groups on the other carbons of the sugar. Okay, so now we have a phosphate group which has formed 2 ester bonds to 2 sugars. Okay, so this is now called the phosphodiester bond. Okay, and this can happen many times. So we could have another one up here. And in this case, it might be cytosine. And again, the phosphate group can make a bond to the sugar over there. Another phosphodiester bond. And thus what we will be constructing, if we carry on in this process, is what we call the phosphate-sugar backbone, or the sugar-phosphate backbone. Okay? So if I just draw this, then this, what we have here, is the backbone. The sugar phosphate backbone, right? So this is the backbone, and what you have sticking out from that backbone is, or are, the nitrogenous bases. Okay, and it's, you know, the sugar, phosphate, sugar, phosphate, sugar, phosphate. There's nothing unique about that. That's just a repeating unit all the way along. But what does give each polynucleotide its kind of unique identity, and the reason why it can act as information storage, is that you then develop a very unique sequence of these nitrogenous bases. Okay, and depending on that sequence, it can give, you know, it can be a way to store information. And how that information is used, we'll look at that later on. Okay? So I hope you can appreciate now that suddenly we have the joining of separate nucleotides, we have the formation of phosphodiester bonds, forming the sugar phosphate backbone, and then we have a particular sequence of nitrogenous bases then developing along that chain of nucleotides. Okay, so let's just finish this off. So this could continue, and we could get continuation up there as well, right? So this could be just one segment of a DNA strand, but that's not the complete structure of DNA, okay? Because what you have is the opposite strand. So just like you got a strand here, you'll have another strand which base pairs with this one, okay? So what will happen is that these bases have groups on them that can form hydrogen bond with another base. Now what happens is that cytosine always base pairs with guanine, and adenine always base pairs with thymine, simply because they have a shape that is complementary, okay? They have OH groups in such a way that when they come close to each other, the partial positives and the partial negatives align with each other. Now this isn't the case between adenine and guanine, and this isn't the case between cytosine and adenine, for example. Okay, but this is the case that whenever you get an adenine base, nitrogenous base, and whenever you get a thymine base, they will align up so that they will form hydrogen bonds between each other. Okay, so if this happens, what you get is you get other nucleotides floating around, and they will base pair via hydrogen bonding. Okay, so if I had another nucleotide, and that other nucleotide had guanine as its nitrogenous base, it will Guys, I was talking for about 10 minutes and just realized that it wasn't recording, so I'm going to have to do it again. But that's life. Struggle is real, guys. Right, here we go again. Right. So, as I was saying, this is just half of the structure of DNA. What it has also, as you know, the iconic diagram shows that there is actually two strands in DNA. And what we're going to look at now is why that is. Okay, why is it that there are two strands? What's holding them together? So, what's holding them together is the interaction that one base can have with another one. Have partial positive and partial negative regions. And what occurs is that if two bases are complementary to each other, i.e. if their positive and negative groups align with each other, they will form hydrogen bonds with each other. Okay? Now, this doesn't occur between cytosine and adenine because the shapes of those molecules are not complementary to each other. Okay? Their negative and positive partial charges don't match up with each other. However, it does occur between C and G, and T and A, and vice versa. Okay, so if I had a G base, so if I had a nucleotide with a guanine base attached to it, nitrogenous base, then it might float around for a little while, but eventually it would, come in the area of cytosine and align with it. It would pair up. The hydrogen bonds between the two would lock that guanine nucleotide in place. Okay, so the other thing you'll notice is that this only happens when the other nucleotide is in the opposite direction. Okay, it's in the opposite direction. That's when the C and the G have a complementary interaction of hydrogen bonds. The other thing you need to know is that C and G form 3 hydrogen bonds between them. Okay, C and G form 3 hydrogen bonds between them. Now, if by chance there happened to be a T nucleotide around, then a similar thing would happen when it lined up with an adenine nucleotide. Okay, in the same way it's flipped around, and when that happens, their partial positive and negative charges line up, and they, between them, form 2 hydrogen bonds. Okay? Let's just get rid of that for now. They, between them, form 2 hydrogen bonds. And in this way, if you have a very long strand, and you do, you only need that one strand, and the other strand will just start forming itself. Now this is very lucky, because if you had one strand, you could make 2. Maybe you could duplicate your DNA, right? Pretty cool. Maybe that might allow organisms to replicate their genetic information and, I don't know, make 2 cells from 1. Maybe that might have been useful to a very primitive cell a long time ago. Anyway, back to this. So you've got the nucleotides lining up, and we've got a G here. So if we had a C nucleotide floating around, it would line up also. Okay? Remembering that the bases have to be in the opposite, sorry, the nucleotides have to be in the opposite orientation for that to happen. Okay, and when that happens, just like that, they'll form 3 between them, 3 hydrogen bonds. Okay, so you've got the nucleotides lining up because of the complementary base pairing, which occurs because of the hydrogen bonding. Okay, this is important. And once you've got the nucleotides lined up, it would just take an enzyme to be kind of following them as they do that and form a an ester bond there, an ester bond there, and an ester bond there, and so on, until suddenly you had 2 strands with complementary base pairing, hydrogen bonding, holding those 2 strands together, and you have 2 strands of DNA. Is it a double helix yet? No. It's a double helix when these 2 strands start coiling, right? Because these nitrogenous bases happen to be very hydrophobic. Okay? 2 strands of DNA like this are exposed to the watery environment of the cell. Okay? But what happens is, because of the coiling, the bases are then shielded from the water. Okay, so the 2 strands coil up like this. Bases are in the center of that coil, away from the water. Okay, very similar to how the hydrophobic amino acids in a protein will be in the core of the protein, away from the surface. Okay, right? So if this structure then coils, then we get the DNA double helix. But this is the, the essential structure of DNA. Okay, how is it adapted, not well adapted, or how is it well suited to perform its function? A, you've got a long sequence of this these nitrogenous bases, and there could be, many combinations of these bases, CAG, GGG, CCC, TAC, etc. So you can store quite a lot of information in a long strand of DNA. Secondly, because it's double-stranded, it's quite stable, which makes it good for storing information over long periods of time. And third, because you've got hydrogen bonding, which is a weak interaction, you can separate them. You can separate the two strands together so that you could allow maybe transcription to occur, or you can allow the replication to occur. Okay, so if you separate the two strands together, so if these were covalent bonds between the two strands, it might have made a very stable molecule, but it wouldn't have allowed the separation to occur when you need to copy information or when you need to copy the molecule. Okay, so it's very useful that you have this weak interaction holding the two strands together, because the two strands can easily then be taken apart for protein synthesis or for DNA replication for mitosis. Okay?